Hey, good afternoon, everyone. I am Tjaart Krüger from the University of Pretoria. I am part of the Department of Physics. So what you will hear today is that I will approach this topic of biophysics from a physics point of view. So let me give a little bit of background about myself. I did my undergraduate in physics. So I have a training in physics, mathematics, and applied mathematics. And it was actually at the end of my undergrad that I started to get a great interest in biological systems. And the irony is that I never had biology during the last 3 grades of my school. I left that. I followed very much the traditional approach that, okay, I really like the physical subjects. I didn't like the subjects like biology because I thought those are the subjects that you have to memorize too much. No, that is not really for me. But I started to become quite interested, and then I realized that biophysics is a pretty new field, and that with my background in physics, I can go into this field of biophysics. So I completed my master's in physics, where I focus on space physics, and then I decided to switch from my PhD to biophysics, and that is a career change that I've never regretted so far. Okay, yeah, if you any questions about that, we can discuss that afterwards. Okay, let me now get to this topic of biophysics. So I do appreciate it very much that I am given the opportunity to give 2 lectures on biophysics, and I hope that you will stay on for both lectures and also be inspired by this relatively new field of physics. Okay, let me give you a brief overview of the 2 lectures. So in the first lecture, I'm going to focus mostly on basic concepts. If you had biology in school, say, up to grade 12 level, then some of this might be a repetition for you, but I thought it's very important to lay out the context before I show you where the physics fits in. So that will be the first hour, basic concepts. And in the second lecture, I'm going to focus a bit more on very specific and selected theoretical and experimental examples. Um, yeah, and those are the main topics: quantum biology, and then some experimental topics: manipulation of single biomolecules, biomolecule-nanoparticle interactions, and super-resolution imaging. But now in the first hour, I'm going to give you a brief introduction to biophysics, and then I'm going to zero in a little bit on the biological cell, just for the context, and then most of the talk is going to discuss aspects of molecular biology. So first, what is called the central dogma of molecular biology, and then macromolecules and the DNA and the proteins. And obviously, I would always try to give you a physics perspective to these systems. Right, so what basically everybody asks who's new to this field is this question: what is biophysics? Okay, let me first start with a negative. So biophysics is not biology and it's not chemistry. And one can see it similarly to if you consider the topic of this, or the subdiscipline of geophysics. This is not geology, just like astrophysics is not astronomy. So what is it? Well, astrophysics is using the principles and methods of physics in the context of astronomy. The same with geophysics, using physics in the context of geology. And likewise, you can describe physics as using the principles and methods of physics to understand how biological systems work. Okay, and very often chemistry is also involved. So one could say that this is a place where physics, biology, and often also chemistry merge. And yes, biophysics is extremely broad, extremely diverse. It covers all scales of biological organization, from the molecular or even the submolecular level, where some topics like quantum biology are introduced, and up the scale to the level of organisms and even populations of organisms, so the full scale of that. What's also good to note is that biophysics is a notable source of innovation. There have been so many innovations that have resulted from advances in biophysics, and especially where physicists were involved, because in general, where you involve a physicist, you um, you can analyze a system from first principles at a quantitative, deep level, and this is also a wellspring of innovation. Okay, and then just a last note, what is the difference between biophysics, biochemistry, biological physics, physics in biology? If you're new to this field, then maybe you don't need to bother about this, but if you have ever wondered about this, this is more than semantics. Um, and I would say, well, my take on this, what I have learned during the past 10, 15 years is that biological physics and physics in biology are basically synonyms, they are the same. They focus more on the physics of a biological problem in the context of biology, whereas biophysics is in general more concerned about the problem itself, the nature of the problem, and then using the principles of physics or often even some chemistry tools to answer or to solve the problem. So it's more, I would say, more focused on the problem itself, whereas the biological physics is more focused on the physics. So you see there's a slight nuance, but I think that's a very important difference. And then biochemistry focuses on the chemistry of the biological system. Okay. Um, yeah, so I just wanted to show you two slides to show you that biophysics is all around us. If you are trained as a physicist, you should actually train yourself to look at the whole world from a physics point of view, just to see physics everywhere you look. And when you look at nature, well, most of nature on the earth is biology. So if you look at this from a physics point of view, you will see so much physics. You'll actually see biophysics all around you. Here are two examples. Why have you ever wondered why fish very often swim in a school, or why birds often fly in a V-shaped formation? Well, the answer is this has to do with energetics. It's more energetically favorable for the fish and the birds to flow in these formations. It's literally built into their DNA to flock together like this. So you can say birds of a feather flock together. Well, we all know this is true, but why from a physics point of view? Well, you can use quite simple fluid dynamics to describe this. This is beautiful physics that is at play here. Okay, this is now at the population level that you can already see the physics. Let's go one step down to the scale of a full organism. And here is an insect that walks on water. And what you see here is, in fact, a physics problem. So this insect is exploiting the special properties of water, specifically the water tension, in order to balance its weight. So this is a pure physics problem that you're looking at here. Okay, so we can go down from the organismal level down to the molecular level, and even the interaction with, in this case, water molecules or some ions, some protons, so we can even go lower on these scales. So this full spectrum is covered by biophysics. And one aspect that makes biophysics really fascinating is, besides the fact that you have such an enormous diversity in in organisms and even processes within each organism, you are also dealing with an immensely broad scale, both along the time axis and along the spatial axis. And this example of a tree is one of the nicest examples, showing you the very broad range of scales. So we start here from a tree. Say it's a very tall tree that you start with almost 100 meters, and you can go down to the scale of a water molecule, which is almost 1 angstrom. So this is, so what it says, 12 orders of magnitude along the spatial scale. Now the temporal scale, if you consider seasonal variations, you're at the magnitude of 10 to the 6, more or less. And if you now go down to the level where a photon is absorbed within 1 femtosecond, 10 to the minus 15 seconds. So this will give you a range of 21 orders of magnitude, which is really astounding. I would say it's an extremely nice playground for a physicist because of this extremely large range in time and in space. And the fact is that with the technology that we have available these days, both computationally and experimentally, we can measure the processes on all these time scales. We can measure literally how a photon is absorbed by a light active, a photoactive molecule. And you can measure up till the seasonal changes. So changes on timescales of years. You can also simulate this, making much faster using supercomputers, all of that. Another aspect I would like to point out is the property of emergence, emergent properties. So this is when a system is more than a sum of their parts. And you see this especially in the context of biology. So if you put some parts together at some hierarchical scale, so say that we put all these molecules together to form, in this case, a photosystem, then this photosystem can do much more than what you would have predicted from the separate molecules. And the same emergence applies at all these different levels of organization of a biological system. Okay, you might have heard that this century, the 21st century, has been called the biological century, just because we have the technology available to solve some of the most complex problems in the universe. And the most complex problems in the universe are in biology. They are extremely complex problems, but we have the tools now to make significant advances in this field. And even if I just look back at the past 10 years or 20 years, the advances are staggering. It's really staggering. It's really difficult to keep track of everything that's happening in this field. I know it's in all of science, but I think it's especially in biology, just because we have the tools available and so many more people are participating in this field. Okay, yeah, here are some two, let me say, testimonials, so quotes from two biophysicists, which I thought might be inspiring to you. I checked actually before the talks, I made this slide a few years back. This link is not working anymore. By the way, the slides are already on the agenda page, but I've made a couple of updates, so I will add the updated versions soon to the agenda page, or actually ask Ketevi to do that. But yeah, you should be able to find these quotations. So I just quickly Googled and I see that this full quotation from which I extracted this, you can still find at this specific link. Okay, so Deborah Feigenson is doing research on DNA origami. So DNA origami is already a very interesting tool. I'm going to introduce it briefly in the second lecture. So for her, it is the immediacy of biophysics that makes the subject so attractive. And yeah, to quote her, it's the potential for impact on human life, the phenomena being close by, she says. I think that if we start to better understand the physical limitations imposed by biomaterials, we'll have fundamental insights into why biology is constructed the way it is. Okay, so for her, it is just that yeah, the immediacy, just to feel that biophysics is literally closer to our skin, and it's easier to find societal applications for that. Whether it's in health or my own research is, one can also link with energy. There are so many applications there. Thomas Krause from the University of St. Andrews in the UK. So yeah, you can actually read this all this text yourself. So it's a bit funny the way he says it, but I think there's a lot of truth in what he says, that he was starting to get bored of telecom. So he had, let's say, a normal job, and yeah, he was getting bored with that. Whereas science and then biophysics in particular, there was just so much to explore for him. So never a dull moment, always something interesting. Yeah, this is also an interesting point of view, as a commentary in Nature Physics from Christopher Jarzynski. And I quote him: biology provides a natural setting for applying and refining the tools of non-equilibrium statistical physics. If an equilibrium state is one in which nothing seems to be happening, then living organisms which grow, move, and multiply seem to be the exact opposite. Just how does a living organism maintain itself away from equilibrium? Processes such as growth and motion arise from intricate networks of chemical reactions driven by chemical imbalances, that is, chemical potential differences. So I don't know how good is your background in statistical physics. So basically, well, the traditional approach to statistical physics is equilibrium statistical physics. That is the easiest. You can derive all the equations that you need. You can make all the predictions. But this is not life. Life is, by definition, in a non-equilibrium state. Otherwise, it is not living anymore. Non-equilibrium statistical physics is really a tough subject. There's so much more to explore in that field of research. And a very good testing ground for non-equilibrium statistical physics is biological systems. And I believe we actually need a few more decades of developing theories before we can really appreciate the full extent of non-equilibrium statistical physics. And then Erwin Schrödinger, I don't know how many of you knew that he had an avid interest in biophysics. One could call him one of the fathers of biophysics. He wrote a monograph, a booklet called What is Life? And it is really worth reading it. Very insightful. And yeah, here's a small extract from this booklet in which he wrote, to avoid a quick decay to equilibrium, a living organism must continually increase the entropy of its surroundings. By digesting food, breathing air, and absorbing photons, living organisms channel or transduce a continual flow of energy that produces the dissipation required to sustain life away from equilibrium. This is just a beautiful perspective from a physics point of view on a biological system. And this is the way I also try to look at these biological systems, and I hope that you will also start to look at them in a similar way. Okay, let me give you a bit of context very briefly. I will start with the biological cell. I will not say very much, actually much less than what you have probably learned at school or what you already know. So there are two main types of biological cells. One so-called kingdom is called the prokaryotic cells, in which you have the bacteria and the archaea, and then you have the eukaryotic cells, and they are differentiated between the plant cells and the animal cells. Okay, so the main differences between these two types of prokaryotes and the eukaryotes is, in the eukaryotic cells contain a cellular nucleus, which contains the DNA, all the genetic material, whereas in a prokaryote, what you see here, this spaghetti-like thing, that is the DNA that just extends over the whole cell. This is actually the main difference between these types of cells. There are more. This is the main difference. The main difference between a plant cell and an animal cell is the fact that the plant cell contains a cell. So no, it's not a membrane. It's the shield. Oh, I forgot the word about. And obviously the chloroplast, which is used by the plant to make its own food, by itself an extremely fascinating process. Okay, let's look at the plant cell just to show you all the different compartments. Now, so actually something else to note, that in the prokaryotes, there aren't compartments in a similar way as in these eukaryotic cells. It's more like just a mix of spaghetti bolognese everywhere. So everything is just everywhere, whereas you have very nice compartments in these two types of cells. Okay, and I like the description of a plant cell as it's actually a mini city. It's often described as a factory, but it's much more than a factory. One can rather see it as a mini city, where this nucleus is like the city hall. That's where the DNA is hosted. You can say all the decisions are made, where everything is programmed, actually pre-programmed how everything should work. and you have the mitochondrion, which is the energy powerhouse supplying all the energy, well, okay, most of the energy for the plant cell. Of course, you also have the chloroplast, which is the solar farm. You can see it like that. You have the endoplasmic reticulum, which is a transportation system. It contains ribosomes for protein synthesis, which you can conceptualize as a factory or a construction site. Okay, then you have this vacuole in the center, which is there for the turgidity control and metabolite storage. So this is like a storage unit. You have the Golgi apparatus, which functions like a post office. It's responsible for packaging and processing of macromolecules. The lysosome you can compare with a garbage company. So each part, which we call an organelle, has a very specific function, and everything works together in synergy. Just want to check the cell wall. Thank you. Yeah, sometimes when you lecture, you just forget very um, um, you just forget some of the words. So thank you for that. Yeah, and if I say anything with which you don't agree, you're welcome to use the chat. I will also, and this is a nice way to interact actually. So a few slides further down, I have some questions for you. I would try to make it a little bit interaction. I know it's really difficult on such an online forum. Okay, so that's the cell. Let's check just a little bit from a physics point of view to appreciate some of the complexity of a cell. And I've taken this figure from the book of Thomas Nordland and I think it's Peter Hoffman, chapter 7, where he's describing the so-called minimal cell, and where they argue what are the minimum components, the minimum number and types of components in order to have a fully working cell. And they've reduced this to basically only these components. Okay, this cell is not going to do that much. It won't be as remarkable as all of the real cells, but at least it can function, it can replicate, it's a living cell, a complete unit. Okay, so then we consider all the processes that form, and we describe all the processes in the form of a simple physics equation. So let's start with the simplest process, diffusion across the membrane. So these small molecules can diffuse across a membrane, and this is the fusion equation, where we have considered only one coordinate. So that is already a dramatic simplification, because in fact we should consider 3 coordinates, but let's just consider that this whole system is spherically symmetric, so we can consider only a single coordinate, say the radial coordinate. Okay, so then you have this diffusion equation, and let's also assume that all the other reactions, so molecules with each other and feedback mechanisms and all of that, can also be described by a similarly simple equation. And you should actually add source and sink terms, but let's neglect all of that for now. Okay, so what you now have is a set of coupled partial differential equations. And I don't know if you have the same a bad feeling about partial differential equations, that they are notoriously hard to solve. So if you have only 2 variables, it's relatively easy. You can do it by hand, or you can give it to you can write your own program to do that. That's pretty simple. Once you get to 3, don't try that to do it by yourself. You should ask your computer program, MATLAB, Python, Mathematica, to solve that. They will do it quite quickly. Yeah, well, I say quite quickly. Even with Mathematica, you might need to go and fetch a cup of coffee, and during the time that Mathematica will try to solve 3 coupled partial differential equations, if you try to do it on your own laptop. With 4, it will take much longer to do that. Okay, now what you have here is that basically every line represents a reaction in the cell. And this is just, appreciate the fact this is an extremely simplified example. And here we have roughly 30 processes. So say 30 coupled differential equations. So I've already said that with 4, well, maybe we can manage with 5. if you have a supercomputer, well, maybe you could try 6, but yeah, you will never get to 30. This is just going to take too long. Okay, now let me show you a snapshot of metabolic pathways that are happening in a typical cell. And just look at all the lines that we have here. I just hope that, okay, there we go. Now this is just staggering. I mean, there are hundreds and hundreds of lines that you have here. And imagine that you have to describe each of these lines with a partial differential equation, and most of these reactions are coupled. I hope that you start to appreciate the complexity of a biological cell. They are just extremely complex. Okay, there we go. Right, this is basically all that I'm going to say about the cell. I would like to zero in on the molecular level, because that is, in my opinion, where some of the most interesting physics takes place. Sorry, before I jump there, let's just very briefly look at another type of cell, the bacterial cell. And here I have a question for you. What is the ratio of bacterial cells to body cells in the human body? Okay, and I have given you here 4 options. You can write in the chat which one you guess is the correct answer. So is there 1 bacterial cell versus 100 body cells? It's a ratio 1 to 10, 1 to 1, 10 to 1. Oops, no, that's not the answer there. Okay, just a few ideas. Now, we're not going to grade you, so even if the answer is wrong, don't feel bad about that. Just guess anything. I would just like to get an idea. Okay, D. Someone says that there are more bacterial cells than human cells in our body. So who else would like to give it a try? Okay, another D. Okay, yeah, so believe it or not, the right answer is indeed D. Now, there are more bacterial cells than human cells in our bodies. It just signifies the interdependence, actually the synergy between all the different types of cells, that we need these bacterial cells for very important cellular functions that have to be executed. So if this is a ratio, does it mean that we are more bacteria than human? Well, this may be more a philosophy question, but I think the answer is quite simple. The average sizes which we deal with here is 2 versus 50. So we are just a bit more human than bacteria. Okay, bacteria themselves are very interesting systems to investigate, and the variety is also staggering. So according to a database, for example, here's a bacterial library, there are more than 30,000 formally named species. So these are only the bacteria that have been identified and studied, but there are many, many more that have not been studied. Okay, let's have a very brief look at viruses. Okay, so what is a virus? This picture always gives me a very nice idea about that. It's like you can compare it with alien invasion. This is like the spacecraft, and there are all the green people that get out. So the virus is a nanoscopic parasite that cannot live on its own. It is dependent on its host. So it goes and it targets a host, and then it injects its genetic material inside the host, and it's dependent on that host to multiply or replicate this genetic material in order to replicate the virus itself. Okay, so there's some debate on whether a virus is a living organism or not, but if you would use the strict definition of a living organism, which should be a a sustained system, the virus is not a living organism because it depends on the host. Okay, and there are really so many viruses on Earth, it's unbelievable. If you would stack every virus end to end, they would stretch 100,000 light years. Yes, there are, by estimate, in order of 10 to the 31 viruses on Earth. Now, this is really amazing. And the diversity is also astounding. So for every person on Earth, there will be enough viruses to study the whole lifetime. You will very easily be able to find your own niche in this field if you're interested in viruses. So how effective are antiviral drugs? We know that antibiotics are effective. They actually kill bacteria, but only bacteria. They don't work for viruses. So in fact, there are, to date, no drugs that can kill viruses. So all the antiviral drugs that have been developed, they serve to prevent growth of the viruses, or to prevent the spread of the virus in your body or in a plant or whatever. So they suppress the growth, but they cannot kill the drugs. Yeah, so if you're interested to go into this field and find the first antiviral drug that actually kills a virus, this is going to be a great revolution. Okay, let's now go to the molecular level, and I'm going to introduce you to the central dogma of molecular biology, which, if you don't remember anything else about molecular biology, just remember the central dogma, so-called central dogma. Well, there's actually also debate whether they should really call it a central dogma or not, but the traditional central dogma is that DNA makes RNA makes protein. Just remember this. DNA makes RNA makes protein. And to expand this a little bit, so DNA contains the full code of life. So that the full code, all the information to make the cell work. And the idea is to make these workhorses, these molecular machines, which are called proteins, to execute all the functions, or basically all the functions in the cell. And it's doing it via this RNA, which is just the intermediate step. So DNA makes proteins via RNA. And actually there are two types of RNA, which I'm going to introduce to you, the so-called messenger RNA and the tRNA, which stands for the translation. Okay, so let's first look at the DNA. You've all seen this. DNA stands for deoxyribose nucleic acid. Okay, and yeah, what's actually contained in this word are these three parts, which are the base, the sugar, and the phosphate. Okay, so this is the base, which is the same for, no, sorry, that's not the same. The sugar and the phosphate groups are the same. So the sugar, okay, you have, for a DNA, you have hydrogen there. For RNA, the structure is basically identical, but you have, instead of hydrogen there, you have a hydroxyl group. Okay, and then we have the phosphate as well. Okay, the phosphate and the sugar together form the backbone, and the base determines the type of nucleotide. Okay, so there are 4 different nucleotides that make up a DNA structure. So this full a molecule is called the nucleotide. And there are 4 different types called thymine, adenine, cytosine, and guanine. Okay, so you can see the sugar phosphate backbone, that is the same, and it's just the base that changes. Okay, and these nucleotides are always ordered in such a way that we start with the side of the phosphate group, and connected to this, well, on this side, you have 5 carbon atoms, and this is why this end is called the 5' end. You start with this side, and the opposite side is then a 3' end, where you end up with 3 carbons there. So always go from the 5' to the 3', and that's also the way in which the transcription and the translation takes place. Okay, how does the bonding work? So these bases always pair up in the same way. So adenine always bonds with thymine, and cytosine always bonds with guanine. Right, so if you have adenine at the one side and cytosine at the other side, you know that, so what nucleotides are going to bind on the other side, because this is always the same, like this. Okay, so you can see again the sugar phosphate backbone. Okay, so for a single-stranded DNA, it's only one part. So you have the sugar phosphate backbone and you have one base there, and the nucleotides are connected via this backbone. Okay, and then you link the second strand, and this gives you then a double-stranded DNA. And since these base pairs pair up, the adenine with the thymine, guanine with the cytosine, when you have the single-stranded sequence, you know exactly how the other sequence is going to be. Okay, and this second sequence always binds in an anti-parallel fashion. So whereas we start on this side with the 5' and we go to the 3', the other strand, we will start with 5' at the bottom or the opposite end, and we'll go to the 3' upwards there. Okay. Yeah, so I've basically already said this, that you only need a single strand in order to identify the full double strand, because when you know this sequence, you know exactly how it will pair up, because you know that the guanine will always pair up with the cytosine, the adenine will pair up with a thymine, and so on. And the same applies to RNA. The only difference with RNA is that instead of a thymine, which you have for DNA, you have a uracil for RNA. But the other three nucleotides are identical, just a small change or a small replacement for RNA. And RNA, you can also have single-stranded or double-stranded, and I hope that you have learned a little bit about this with the vaccines that have been developed for COVID-19. So most vaccines are based on the messenger RNA, where they will then show such a sequence of the nucleotides, and that then identifies the full RNA strand that makes up this vaccine. Okay, who knows what is this? Let's see, I don't like these online platforms because people can't shout out. But does anyone know what I'm showing here? Okay, this is probably the most important diffraction pattern in the history of mankind. This is the diffraction pattern of the DNA molecule. Okay, and this was published in 1952. 10 years later, the Nobel Prize was given for that because it was just a revolution. And if you know diffraction patterns well, you can see that this is a beautiful diffraction pattern and you can get all the information very easily. So this angle is the tilt of the helix you see here. 1/p is the distance of one complete turn of the helix, and 1/h is the distance between the bases, the nucleotides. Okay, and here you also see the numbers there. So they have perfectly resolved this and perfectly found all this information which still stands today. Okay, let's get further with the central dogma of molecular biology. So we've looked at the DNA. Let's now look at how this transcription process works. Okay, so remember the days of photocopying textbooks. Well, that was, yeah, I'm getting old. It was still during the time that I studied that a lot of photocopies were made. So say that there's a biology textbook and you need it for 3 different classes, and you make 3 copies from chapter 1 for the microbiology class. You make 20 copies from chapter 5 for the genetics class, and you make 6 copies of chapter 7 from the zoology class. Okay, so this is your master copy, and then you selectively want to copy certain portions of this master copy? Well, transcription works in a conceptually identical manner, and this is where RNA comes in. So RNA, messenger RNA, um, is normally a relatively short strand. This is a full strand that is needed for the protein, but remember that the DNA is an extremely long molecule. Okay, and it contains all the genetic material for all the proteins, etc. But one strand of messenger RNA will translate or actually transcribe the information for a single protein. So the way it works is that an enzyme, which is a type of protein, will come and unzip part of the DNA, just the part which needs to be exposed for the messenger RNA to read off this genetic information, and then it will go out of the nucleus and then give the information over to the tRNA, which will then make the protein. Okay, so here you also see this so-called RNA polymerase. Okay, and the messenger RNA, which only goes to this so yeah, so these two strands of DNA, the one going from 5' to 3', that is called the sense, and the other one is called the antisense, the matching one. So the messenger RNA will only go to the antisense so that its genetic information will be identical to that of the sense strand, the coding strand. Okay, so once it has copied the genetic information there, it goes out of the nucleus, it goes to a ribosome where a protein will then be synthesized. Okay, and here are all the different proteins that are being, the amino acids that will be synthesized, and there are 20 essential amino acids. So how can we use only 4 nucleotides to make or to have the necessary information to translate 20 essential amino acids? Well, it's very simple. If you have a single letter code, there are 4 possibilities with 4 nucleotides. If you use 2 nucleotides at a time, you have 16 possibilities. If you use a triplet, so 3 nucleotides at a time, you have 64 possibilities. Okay, so this is exactly how it works. It's called the codon system, which is a 3-letter RNA code, which would correspond to a single amino acid. Okay, and this is the universal code for life. Okay, and yeah, so there are many more than 20. This leaves some space for a stop and the start codon, and there's also some redundancy, so different codons that can translate to the same amino acid, which is then also used to reduce errors. Okay, so here are all the different amino acids and all the different triplets that relate to that. And I know that I'm going a bit fast over this. And yeah, I'm also going to skip this. I would like to encourage you to watch this YouTube video if you have never been introduced to the central dogma of biology. It is just fascinating how it works. All these different molecular machines that work together in order to code proteins, the molecular machines. Okay, let me now go over to the peptide chain. So peptide chain, that is a part of a protein, and very similarly to DNA, it contains this backbone, okay, which runs from the so-called N-terminus to the C-terminus, and then there are all these functional groups, and the backbone is always the same, but the functional group, that will determine the function of the protein. And here are a few names of such a strand of amino acids. Okay, so that brings us to proteins. So proteins are really fascinating systems because they are responsible for basically all the functions that happen in all the cells of all organisms. So they are really the workhorses of the cell. And proteins are molecular machines. They are nanoscaled machines. Each of them has a different function. and they're just marvelous systems. Some people call them the hydrogen atom of life just because of their importance in biology. In the human body alone, there are more than 100,000 types of different proteins. And the reason that I can't give a number, a full number here, is that nobody knows yet how many types of proteins there are in the human body. Some say there could be up to 1 million different types. Nobody really knows. Only a small fraction of proteins have been identified and sequenced so far. Okay, so what is a protein? A protein consists of amino acids. It's a chain of amino acids. It consists of a carboxyl group on the one side, amino group on the other side. These two form the backbone. Okay, and then there's the side chain, which gives the protein a certain function. So the side chain is the one that varies from protein, or sorry, from amino acid to amino acid. These two groups stay the same. So the amino group, you have amino, and the other group is you can call it an acid. So this is why we call this an amino acid, because these two groups are the same for all the amino acids. It's then just the functional group, the side chain, that differs for all of them. Okay, here's again all the essential amino acids. There should be 20 of them. I see there's 21. But yeah, that's because in the human body there are 20 amino acids. In some other organisms use some other proteins that use some other amino acids. Yeah, so I've already said this. So all these amino acids differ just because they have different functional groups. And some of them are more acidic, some of them are more basic, some of them are, so the charges are just completely different. Some of them are charged, as you see here. Some of them are more hydrophilic or polar, some are hydrophobic, and these properties all determine exactly how a protein will fold up and also how it will interact with other molecules. Okay, so we've already seen this. I'm going to skip over this peptide bond just to bind different amino acids. Okay, let's go to the secondary structure. So you have the primary structure, which is just the sequence of different amino acids forming a long chain. And then there are two distinct types of secondary structures, which often form so-called alpha helix and the beta helix. So beta sheet. Okay, and they are stabilized by hydrogen bonds. And hydrogen bond is a relatively strong interaction. So these secondary structures are relatively stable. Hydrogen bond is obviously a kind of dipole-dipole interaction. So you can already see the interesting physics that is also involved only in the structures. Okay, and there's a beta sheet, which also contains a number of hydrogen bonds. Just see. Okay, so these are two types of secondary structures, and then eventually the protein will fold up into the tertiary structure. So that's the most compact structure, three-dimensional structure, and the structure determines the function. So it means that each sequence of amino acids will have a different structure just because of the nature of the amino acids, the hydrophobicity, or how water attractive or water repellent it is, whether it's charged or not. Okay, and this outer surface of the protein will then determine how it's going to interact with other molecules, including water. So this shape is what determines the function of the protein. Okay, here's one example. The shape of the hemoglobin is such that there's a pocket inside which is just big enough for this heme molecule to attach. Okay, and the function of the heme molecule is that it attaches oxygen to transport the oxygen in your blood. So there are a lot of these hemoglobin proteins inside your blood to transport the oxygen through your blood. Okay, I also wanted to say a word about protein folding, which is a fascinating area of research. A lot of physicists are also involved there. So this is the question: how do you determine the three-dimensional structure or the fold, three-dimensional fold or the tertiary structure of a protein from the primary structure, which is the sequence of amino acids? And this is not a trivial question. There are a lot of driving forces that you have to take into account. Most important, in order of importance, more or less, are the hydrophobic interactions, hydrogen bonds, and other van der Waals interactions, because the first two are actually also a type of van der Waals interaction. Electrostatic interaction, and then there can also be disulfide bridges if there are specific amino acid or amino acids in the proteins that will give rise to that. And all of these are opposed by the conformational entropy, which is a force that would prevent the protein to fold up into a three-dimensional structure. Okay, and so the system has to get a balance between all these forces, and such that the entropy is the lowest. So it's in the most ordered configuration. Okay, and misfolds happen. They can actually happen quite frequently, and they are actually other structures in your cell that also determine whether misfold happen or not. And if there's a misfolded protein, it will be destroyed. But it happens that misfolded proteins do not get destroyed, and they then would often aggregate, and that can lead to severe diseases, for example, Alzheimer's and cystic fibrosis. Okay, so to determine the tertiary structure from the primary structure is not an easy game to do. And we can appreciate that from Leventhal's paradox, which he framed about, what was it, about 60 years ago. So imagine, and this is also a very simplistic scenario that is sketched here. So imagine that there was only a single bond between each amino acid in a protein of 101 amino acid residues. So you have a sequence of 101 amino acids, and that is a small protein. Okay, so imagine then that there were only 3 possible configurations around each of these bonds, which is a very large simplification. So this means that the protein could adopt 3 to the power 100 different conformations. Now, if the protein is able to sample 10 to the 13 different bond configurations per second, which is realistic, then it would take 10 to the 27 years to sample all possible conformations of the protein. And this is a simple protein described in a very simplistic way. So you can see that this is definitely not the way it works in a cell, because proteins fold up typically within a couple of milliseconds, which is pretty fast considering all the different forces that are at play here. Okay, proteins are represented in different ways. You have the space filling diagram, ribbon diagram, surface diagram, and each representation serves different purposes. It depends on what you would like to emphasize. Okay, basically in the last part of this lecture, I'm going to focus on a few protein functions. So proteins are the workhorses. It means that they need to execute specific functions, and each type of protein has a different function, and there are many, many different types of functions. Here is a list of some of the most important ones, but there are many, many more. So you get a whole subgroup of proteins called enzymes, and they serve to catalyze reactions. And then you have proteins that regulate gene expression, others that are responsible for building structures, so getting building structures or giving strength to structures. You have transport proteins that are responsible for transporting molecules or ions across the membranes or to different cellular compartments. You have proteins that are involved with communication, like receptors, or responsible for signaling. You have proteins that are responsible for storage of specific molecules, and others that are involved with defense. You have motor proteins, you have light harvesting proteins, You have the reaction center that converts light into chemical energy. So such a vast range of functions. And I would like to show you just a few of them so that you can appreciate some of that. Okay, here is one, an antigen that is involved with defense. Okay, so so an antigen will target a foreign molecule like a virus, bind to it, and then take it to a site so that the immune system can destroy it. Here is a pretty marvelous protein. Well, actually, different proteins that are involved here. So this yellow protein there is called insulin. And you may know that insulin is very important in your blood because it regulates the amount of glucose in your blood. And how this is regulated is through a complex chain of events. So this insulin, which is a relatively small molecule protein, will bind to a very big protein, which is called the insulin receptor. And once this insulin binds, this insulin receptor will change from this V formation to a T formation. Okay, and here are the electron micrographs that show that it really happens like that. And when this structural or conformational change happens, there's a cascade of events that will ultimately lead to such a transmembrane protein that will appear through the membrane, and then it will pop a glucose molecule into the bloodstream. So this is really marvelous. Okay, aquaporins are also quite marvelous systems. So in basically every cell of every living organism, there are a lot of these aquaporins, which are water channels. So they channel water molecules through the membrane. Okay, and only water molecules. It means that regardless of how filthy the water is on the outside, it will purify the water. So these are actually water purification systems. And if you consider, for example, a marine fish, a fish swimming in the ocean, so the water on the outside of the cell is salty water, and these aquaporins will then desalinate the water by allowing only the water molecules to pass. And this also happens at a significant rate. Well, just consider that in an average human has about 40 trillion cells. And then these cells are packed with these aquaporins. So it's just a staggering amount of aquaporins that are in the universe. Here's another transport protein, and this is really a fascinating system. It is called the ATP synthase, and it is a proton pump. So it is actually a pump, a molecular pump. So it uses protons on the inside of the membrane. So protons are then sucked in on this side. It will go around via the backside to this side, and then it will go to the upper compartment. And when this proton goes around, it creates a torque. So that this is like a shaft that will then turn around. And for one revolution of this shaft, ADP will turn into ATP. So this is a molecule that contains two phosphate groups, and then a third phosphate group will bind to it. And ATP is the universal currency of energy in biological systems. So human beings consume the energy of about 70 kilogram of ATP per day. This is really a staggering amount. And one ATP is formed by 3 protons that create this torque. Really a lot of nice physics in here. So, and yeah, what is also fascinating about this machine is the speed with which it revolves around the axis here. So they found ATP synthases in some systems that can go up to speeds of 42,000 RPM. 42,000, do you know how fast that is? If you compare it with the engine of your car, which is about 1,500 for an average car. Racing cars can go up to about 12,000 RPM. This is 42,000 RPM. It's really amazing. Okay, so we consume about 70 kilograms of ATP, and about 1/3 of that is used by this so-called sodium-potassium pump, sodium-potassium pump. Okay, actually all these three pumps, well, these two on the opposite sides are channels, the potassium channel and the sodium channel. All three of these are inside the membranes of nerve cells. So whenever you have to send something and a signal has to be sent to your brain, or when you are thinking, then nerve cells will fire, and they will fire electrochemical potentials. Well, electricity will flow through your body. There's really so much interesting physics that's going on there. Okay, and the electrochemical potential is driven by these two channels, the potassium pump and the sodium pump, and they work by means of a charge gradient on both sides of the membrane, just like the aquaporins also work by means of forward osmosis, or the charge gradient, the potential difference on both sides. These also work in that way. So by transporting potassium to the outside of the membrane and sodium to the inside, an action potential is created, and this is transported through the nerve cells to the brain. Okay, and then just after such a signal has been fired, the concentrations have to be neutralized again, and this is done by means of this potassium-sodium pump, which then consumes ATP in order to do that. Okay, so if you think a lot, you're going to consume a lot. So you might consume, well, more than 1 kilogram of ATP throughout this talk. That could be quite possible. I see that it's about 1 hour now. Okay, I'm almost done with this lecture. I just want to show you a last type of function, which is also quite fascinating system, ferritin, which stores iron. It has a big cavity on the inside, and iron will go through some of these channels, and then they will bind to the inside. So this is a big, you could say, a storage container for iron for the body. So whenever it will need iron again, it can just take some of the stored iron and then use that. Okay, is it fine if I complete this? I think I would just need a couple more minutes.
Go ahead, yes.
Okay, thank you. Okay, one other aspect I would like to highlight about proteins is that they are dynamic structures. So life is dynamic. Life is about motion, about function. And proteins, or many types of proteins, switch between different functions. For example, here we have a cartoon of an enzyme which will switch between an inactive conformation, and upon binding of an activator molecule, which in this case is glucose, it will then switch to the close conformation, which is then the active conformation, and then it can go and execute a function. Another example is this huge protein system called phycobilisome, which is the main light harvesting complex of cyanobacteria. So the photosynthetic activity, or the light harvesting activity, is switched on and off by this small protein here, which is called the orange carotenoid protein. And this is a photoactivated protein. So it means that it needs blue light. Well, this orange carotenoid protein needs blue light in order to become active. And once it's activated, it will bind to the core of this phycobilisome and then switch its activity off. And we've actually measured this at the single protein level a couple of years ago, where you can see here that the emission, which here stands for the function of the phycobilisome, is switched off upon binding of the OCP. And here's an example where it switches back on again. Really marvelous that this can be quantified at the single protein level, showing the dynamicity of these systems. Okay, and a very last example here, we will then step back a little bit, look at a membrane that is here, in this case, packed with membrane proteins, actually bacterial light harvesting complexes of different sizes. And membranes are also dynamic systems. They can also change their shape, and when they change the shape, the curvature is responsible for self-assembly, so clustering. So you can say some metascale or mesoscopic effects that happen here. And this has actually been modeled quite well using the Helfrich elastic energy. So you can, actually all these systems can be modeled in one way or the other. It's just a way in which you can then model the curvature, and from that coarse-grained model using spheres of different sizes, you can see how this self-assembly actually happens. Okay, yeah, 2 acknowledgements for this lecture. I have stolen a few slides from a colleague in the Department of Biochemistry, Genetics and Microbiology at the University of Pretoria. Then I've also obtained a few slides from the Protein Data Bank educational portal, some of the protein slides there. And I leave some food for thought for you. And here are also a few animations which I just find incredible, getting a sense of the inner life of the cell, everything that's happening there. And I hope that if you look at that, you can just see all the physics that's taking place when all these molecular machines interact with one another. Okay, so this concludes the first lecture. Is there time for questions now? Or shall we first take a break? How shall we do this?
Yeah, typically we have half an hour break, but then I would suggest indeed to take maybe a few questions if there are some now, because it's so interesting. So
yeah, sure, definitely.
We don't have any in the chat box, but please don't hesitate to speak up. So maybe, yeah, Kayode, do you have a question?
Yeah, thank you very much for the lecture. I just want to know, how do you normally interpret all those protein folds for you to know this is a primary protein, tertiary, and all those things like that? How do you get to interpret them?
I didn't fully understand your question. You're talking about potentials, and then the primary and tertiary, are those
I mean the
structures of
protein
protein?
folds, all the protein folds. How do you get
Thank you.
to interpret the results?
How do you measure or how do you interpret, Kayode?
Yeah, including the measure for you to know this is a primary protein. Especially, I could see somewhere in red color, some in white, how they combine for you to be able to understand the results there.
Okay, so those colors are just an indication of charges. So whether the side chains are positively or negatively charged. Okay, you have the primary sequence is just a chain of amino acids. Okay, and this will fold, well, you will have some secondary structures, which are the alpha helices, beta sheets, and the fully folded structure. So the most compact form of the protein, that's called the tertiary structure.
Maybe if you could open one slide and use it to explain that way.
Okay, yeah, let me go back. I know there's a lot of information that I've given, and it might have gone quite fast. Are you referring to this slide?
I think that's an example.
Okay. Yeah, so the red ones are the hydrophobic amino acids. Hydrophobic means water repelling. So these are polar, well, okay, these are molecules that have a polarity, like plus on the one side, minus on the other side. Yeah, so they will, no, sorry, it's the other way around. So when they are plus and minus, they will actually attract the water molecules, because water is also a polar molecule. So those are the hydrophilic ones, that are the polar molecules. Okay, you have the charged ones, they are shown in yellow. Hydrophilic ones, which are water attractive, so they will attract the water molecules. Hydrophobic ones are water repellent. Okay, so on the whole surface, you see that there is so much heterogeneity, and this determines the function of the protein. For example, which other molecules it will bind, or whether it will bind to another protein or to DNA or whatever it has to do. Does this answer your question?
No. My question is, for example, if we don't have those labelings, if I have the picture only, how do I interpret that?
Oh, how you do that without the labels? Yeah, well, there's software that can help you with that. Yeah, because every functional group, so every side chain, you can give a hydrophobicity gradient, or how, you can get a very good estimate of exactly where the charges are. You can even do it from first principles by doing quantum chemical calculations, which will also tell you exactly how the charge cloud is distributed over all the side chains. And then you do this for all the side chains. And in fact, you would then also have to take into account their interactions. So it can become pretty complex. That's why you need good software in order to model it well. But it can be done to a quantitative level. Does that now answer your question?
Yes, but I will still, maybe I will contact you later for more information.
Okay, sure, yeah. We can definitely take the discussion further.
Good point.
Thank you very much.
And maybe indeed to as well develop on that, because it was like for the theoretical part, so to try to understand how also through calculation everything can be observed, but how to measure it. So this is one of the sides which is interesting, because I'm working with the neutron source, so that's one of the things, or with light source. So in the future they're going to be as well, hopefully, this African light source. So there will be the way as well to better understand how with protein crystallography as well, there could be a better understanding and complementarity of those software. So I guess that this is really like a huge field, as you said, the atom, more or less the hydrogen atom of life. I mean, it's wonderful for the protein to try to understand everything that is ongoing. And we'll have to leave the room. Apparently, we're all pushed out. I will still put the presentation by Adayona, which would be as well quite interesting for all of you, because for the ribosome, so she has been explaining everything on how it works. So then I put here, look, so that you will have it for later on. Here, hopefully I'll do it.
I've really just scratched the surface of selected topics. There's really so much.
Yeah, so see this as well, because you had a lot of YouTube, so maybe this is going to be a complement, ribosome action, because we had the chance to have her as well speak, crystallography. So this is a wonderful presentation that will teach you as well a lot, and great movie.
Yeah, thank you very much for that.
I really like it also, and I will check your YouTube so that we can disseminate. I mean, this field is wonderful. It's not only multi-physics,